Team number 6, Campylobacter and Vibrio cholera. Plan of the team present on this slide. Campylobacter species cause both diarrheal and systemic diseases. They are motile, non-sporing, microaerophilic, curved gram-negative rods. Human pathogens fall into two major groups, primarily diarrheal disease, caused by Campylobacter jejuni and accounting for 90% of total cases, and others such as Campylobacter coli, Lari, and fetus, and the extraintestinal infection caused by Campylobacter fetus. And the culture characteristics of Campylobacter disease. Campylobacter species. The culture media for Campylobacter are as follows: transport media and selective media. And transmit media, if delay is expected, transport media such as carrier media can be used. It survives for 1-2 weeks at 4 degrees Celsius. And selective media, feces or rectal swab are plated onto selective media such as Scarro's selective media. This is and the Basler's selective media, and the Campy selective media contains lyse blood agar and vancomycin, polymyxin B, and the cefalozine. And the culture conditions: inoculated plates are incubated at microaerophilic condition, 5% of oxygen and 10% of CO2 and 85% of nitrogen, and the growth at 42 Celsius degree. And thermophilic Campylobacter species, Campylobacter jejuni, coli, and Lari, can be differentiated from Campylobacter fetus, which is non-thermophilic. And after 5 days of incubation, characteristics effuse droplet-like colonies are produced, which can be further subjected to biochemical tests for species identification. And the biochemical identification: Campylobacters are oxidase positive and catalase positive, and catalase positive, and nitrate producers and don't ferment sugars. And the Campylobacter jejuni has two subspecies, jejuni and doli. The subspecies jejuni can be differentiated from the latter by nitrate positive, hyporate hydrolysis positive, and grows at 42 degrees Celsius. And epidemiology: Campylobacter species are zoonotic, found in the intestine of many animals and household pets. Mode of transmission: by raw or undercooked food products through direct contact with the infected household pets, and travel to developing countries. Campylobacter jejuni infection is common among children, and the incidence peak during summer and early autumn. And Campylobacter in humans: most cases of human Campylobacteriosis are presented in isolated episodes, but outbreaks are also observed. Gastroenteritis infections caused by Campylobacter species occurs in humans by oral via through consumption of contaminated food of animal origin or by contact with pets or production infected. It can also occur by consumption of other foods such as unpasteurized milk or contaminated water. How not to feed via transmission can still quote the direct transmission from individual to individual, but is not considered an important mode of transmission in industrialized countries. And the pathogenesis of this disease: molecular and cellular features of the innate immune response to Campylobacter jejuni in humans present on this slide. Campylobacter jejuni circumvents the mucus layer in humans and interacts with the intestinal epithelial cells, causing interleukin-8 production. And Campylobacter jejuni binds to and it is internalized by epithelial cells. The induction of interleukin-8 causes the recruitment of dendritic cells and macrophages and neutrophils, which interact with Campylobacter jejuni. These interactions results in a massive pro-inflammatory response and increases in the corresponding cytokines. And the clinical manifestation of Campylobacteriosis. Campylobacter infection commonly presents as an acute gastroenteritis that is characterized by inflammation, abdominal pain, fever, diarrhea, with an infectious dose as low as 800 bacteria. The incubation period that precedes the development of acute diarrhea is 5 days, and also the disease is typically resolved in 1 week. Symptoms can last for up to 2 weeks. Epidemiological studies of Campylobacter infection indicate that there are 2 disease manifestations which are dependent on socioeconomic status. In developed world, Campylobacteriosis manifests as bloody diarrhea with mucus and is usually self-limiting. In the developing world, watery diarrhea predominates and the infection is more frequent among children, which might naturally vaccinate them against becoming infected as adults. And the laboratory diagnosis of Campylobacter pathogen. This gram staining of smear of feces may show, this is gram staining, may show curved gram-negative bacilli. This is bacilli 1.5 to 5 micrometer diameter, appearing resembling vibrio, or S-shaped or spiral gull wing shaped, and dark ground microscopy demonstrates the darting motility of the bacilli. And the Vibrio cholera. Vibrio cholera is curved or comma-shaped rods gram-negative motile by means of single polar flagellum. As forms of spirals may be seen due to 2 or more cells lying end-to-end, and they are frequently highly pleomorphic. It has somatic antigen, and Vibrio cholera can be grouped into more than 200 serogroups or serovariants. O1 O139 serogroup and non-agglutinable Vibrio. Based on cell requirement, Vibros can be classified into non-halophilic and halophilic Vibros. And the culture characteristics of Vibrio cholera. Culture medium to inhibit the commensals fecal specimen should be inoculated simultaneously onto enrichment broths and selective media. Enrichment broths, they are incubated for 6 hours, and selective media, stool specimen is directly inoculated onto a selective media, and the plate is incubated at 37 degrees for 24 hours. And this is TCBS agar. It contains thiosulfate citrate, bile salts as inhibitor, and sucrose, and has pH of 8.6, and bromosimol blue and simol blue are used as indicators, and this is widely used at present. And Vibrio cholera and other sucrose fermenting Vibros produce large 3 to 5 millimeter yellow colored colonies. This is colonies, and yellow colored colonies, and whereas sucrose non-fermenters produce green colored colonies, and through it is inhibitory to most of the bacteria. Some strains of Aeromonas, Proteus, Enterococcus may occasionally grow. And next is hemodigestion. Vibrio cholera produces hemodigestion on blood agar. This is blood agar. On blood agar, which refers to the non-specific killing of blood cells by metabolic byproducts of bacteria. This can be seen on a blood agar plate as the medium surrounding the main inoculums turns green, but there is no change around single colonies. It grows optimally at 37 degrees Celsius, and growth is better in alkaline medium. The optimum pH is 8.2, and sodium chloride stimulates the growth. However, higher concentration of sodium chloride are inhibitory. And the next medium is McConkey agar. This is McConkey agar. When not sure about the type of enteric pathogen present in feces, McConkey agar can be included in the panel, and it is mildly selective. It also supports other enteric pathogens such as Shigella and Salmonella. Colonies of Vibrio cholerae are translucent and pale, which may become pink on prolonged incubation due to late lactose fermentation. And the biochemical properties, this is catalase, catalase positive and oxidase positive. And the next biochemical properties is indole test positive of Vibrio cholerae, and citrate test variable, and urease test negative, and nitrate reduction test is positive, and Vogelspore scour test positive for L-Tor Vibrio, and negative for classical biotype. And the epidemiology of this disease, first is possibly hypertransmissible virus. If ingested with a few hours, may enhance direct transmission in crowded unsanitary conditions. And virus colonize copepods, zooplankton, or crustaceae, and survive in a dormant state. And next is spread in homes, days, base, or via ship ballast. And next is ingestion of enough virus to overcome gastric acid and mucosal host defenses. Fewer needed if ingested with buffer or certain foods. And the next process is Vibrio-tox R gene is activated in the small bowel to increase expression of TCP. TCP is toxin co-regulated pilus colonization and the cholera toxin secretion that activates adenyl cyclase and chloridium secretion, and Vibrio neuraminidase and mucolytic enzymes enable motile fibers to hemotax to the mucus. And the next process is voluminous watery diarrhea, severe dehydration, shock, and death. And the mechanism of action of Vibrio cholera toxin, this on this slide present of mechanism. And adhesion and colonization. The next step is pathogenesis. It is adhesion to the intestinal epithelium, which is facilitated by a special type 4 fimbria and called toxin co-regulated pilus. It is so named because the synthesis of toxin co-regulated pilus, cholera toxin, and the several other virulence factors are co-regulated by a single gene called TOX-R. And cholera toxin, once established in a human small intestine, the organism produces a powerful enterotoxin called cholera toxin. It resembles heat-label toxin of Escherichia coli in its structure and function, but it is more potent than the latter. Mechanism of action of this toxin. The toxin molecules is about 85 kilodalton, consists of 2 peptide fragments A and B. Fragments A is binding fragments and is pentameric, contains 5 subunits, each weighing 11 kilodalton each. It binds to GM1 ganglioside receptors present on the intestinal epithelium, following which a fragment is internalized and cleaved into A1 and A2 peptides. And fragment A2 helps in tethering A and B subunits together. And fragment A1 is the active fragment, causes ATP ribosylation of G protein, upregulates the activity of adenylate cyclase. A result is the intercellular accumulation of cyclic adenosine monophosphate, and increases in cyclic adenosine monophosphate leads to in small intestine cyclic adenosine monophosphate inhibits the absorptive sodium transport system in villus cells and activates the secretory chloride transport system in crypt cells, which lead to the accumulation of sodium chloride in the intestinal lumen. Water moves passively into the bowel lumen to maintain osmolality, which leads to the accumulation of isotonic fluid that results in watery diarrhea. Loss of fluid and electrolytes leads to shock due to profound dehydration, and acidosis due to loss of bicarbonate. And the clinical manifestation of cholera, this is Vibrio cholera O1 or O139. Infections produce a range of clinical manifestations such as asymptomatic infection, 75% of cases, and a mild diarrhea or cholera, 20% of cases. And the watery incubation period varies from 24 to 48 hours. The usual manifestations include watery diarrhea, water stool, and watery diarrhea. Cholera characteristically begins with the sudden onset of painless watery diarrhea that may quickly become voluminous. And rice water stool. The stool is typically non-bilious, slightly cloudy and watery with mucus flecks and a fishy inoffensive odor. And being non-invasive, there is no associated blood or pus cells in stool. It often resembles the water in which rice has been washed. And vomiting may be present, but fever is usually absent, and muscle cramps may occur due to electrolyte imbalance. And the complications are directly proportional to the fluid loss, which results in loss of body weight. And laboratory diagnosis, direct microscopy we use, and the culture method we use, culture smear and motility testing, and biotyping, serogrouping, serotyping, and antimicrobial susceptibility testing we use. And gram staining on this slide, Vibrio cholera gram stain. They are gram negative. They have one flagella, monotrichous, and curved, comma-shaped gram negative rods.